Welcome back. Part 2. We're continuing on. I hope you drew your DNA structure. What do you remember? And then I think that this will all fit in. Again, I only have a couple things that I think are new to you. Regardless of where you took Bio 1, I think you've had all the rest of the stuff. I want to talk about the structure of DNA. DNA is made out of nucleotides. Those are the monomers, or the building blocks that build in a polymer, a chain of DNA sequences. Nucleotides are made up of 3 building parts. There is a phosphate group, and it's not an atom of phosphorus. We'll look at it a little bit in more depth in this class than you did in other bio classes, I think. This is a functional group that is acidic. It is charged. This is PO42 negative. This is attached to the sugar that makes the basic structure of a DNA nucleotide. This sugar is a 5-sided sugar. It's a pentose. It has 1, 2, 3, 4, 5 carbons on it. And it has, I'm going to put it down here, 1 OH and 1 H stuck to the side. This flat side, or what we call the 3 side of the molecule. This sugar is called deoxyribose. Os tells you it's a sugar. Just like, I was thinking enzyme, just like ACE tells you it's an enzyme. So deoxyribose is a sugar. And then the nitrogenous base, this is where variation occurs. These are T's or A's or G's or C's. There's no G on here. And the adenines, the thymines, the guanines, and the cytosines that make up the variable regions that actually hold on to the code. The nitrogenous bases are either pyrimidines or purines. Pyrimidines are made of 1 carbon-nitrogen ring, and you do not need to memorize the structure here. I just want you to see that they're made of 1 ring. They're smaller. You here in pyrimidine, you see the Y. Cytosine and thymine are both pyrimidines. They have Y's in their words too. Uracil, it doesn't have a Y, but it's also a pyrimidine. Pyrimidines always pair with a purine. Purines are larger than pyrimidines. They're made of 2 nitrogen carbon rings. Again, I do not care about the structure for these things, but do notice that they're larger and made of more rings. I'd like you to notice that guanine and adenine are not that different, but they are different than each other. They have different types of functional groups, so they have different functions. The way I remember purines is they are pure as gold. A and G are the purines. DNA sugar is a deoxyribose. Deoxy literally means to remove an oxygen. And I want you to compare this just briefly to ribose down here. Ribose is the sugar that we find in RNA, ribonucleic acid. Ribose has a hydroxyl and another hydroxyl, or 2 OHs. Y'all have had enough chemistry that I can hydroxyl, right? Yeah. So they have 2 hydroxyls here. Deoxyribose has had one of the oxygens removed from its 2 carbon. It's been deoxied. Deoxyribose, although the word is longer, is actually a smaller molecule. This affects their function substantially. It's going to affect how we can build onto the structure of these, and also what kind of molecule we're going to use this, how we're going to use these different molecules. So this matters. This matters. Nucleotides are the monomer of DNA. They have a sugar, a phosphate, and a nitrogenous base. Nucleosides are just the part without the phosphate. Sometimes we're going to talk about nucleosides. Sometimes, mostly, we're going to talk about nucleotides. Nucleosides are here. This is a nucleoside. And I don't think we revisit them for quite a few lectures. This is a nucleotide. I think you've had enough chemistry at this point to know that when I change the molecular structure, it completely changes the molecular function. Removing the acid from this and not having it attached really changes the way it acts in solution, but it also changes the way that it's going to act with the machines that we're going to try to use to analyze it. Because this is uncharged, it's not an acid. This is charged, it's an acid. So 3 versions. I don't have a picture of this one. Nucleotide triphosphate is when I have, sorry, I said that wrong. Nucleoside here, triphosphate, is when I have a nucleoside with 3 phosphates attached. We are going to use this to assemble the DNA when I am building a new chain of entire monomers, not binding A's and T's, but building the monomers along the rails. My building blocks are going to be nucleoside triphosphates. These 2 extra phosphates we're going to use in the chemical reaction to cause them to form a chemical bond to the other nucleotide they are bonding to. Nucleoside diphosphates have 2 phosphates and a nucleoside. Guess what a nucleoside monophosphate looks like? It's a nucleotide. So this is a nucleoside monophosphate. It's got 1 phosphate on it. That's a nucleotide. Don't overthink this. It's 3 phosphates, 2 phosphate groups, 1 phosphate group. Nucleoside triphosphates are the molecule that we actually use during nucleic acid synthesis. So when I am elongating, making longer my strands of DNA, that's what's floating around in solution, and that is what the enzymes that build the DNA are going to grab and build into my growing molecule. In order to power some of this and some of the other functions in your body, you use a molecule called ATP, adenosine triphosphate. You also use a molecule which you may or may not, depending on the courses you have, talked about a lot, guanine triphosphate. These are exactly what you think they are. They are like the building blocks in DNA. They have 3 phosphate phosphate groups tied to them. If you remember the functionality of DNA, DNA stores its energy in a way by having that 3rd phosphate in a really spring-loaded bond. So as that phosphate is removed, it can be used to change the function or the structure of another molecule and thus transfer energy. This terminal, the 3rd phosphate, is involved in energy transfer. They are going to help us build bonds between nucleotides. Those bonds are called phosphodiester bonds. They link one monomer of a nucleotide to another monomer of a nucleotide. And there's a lot of vocab-y kind of stuff, and I guess molecular vocab. I will tie this all together as we go into DNA replication. I want to have it in your head right now. That's why I didn't make it red. It's not a vocab word yet, but it will be. Chargaff was a researcher that was working, trying to understand how DNA's, if it was the transformative factor, how does its structure show that? And he worked by isolating nucleotides from each other in a lot of samples of DNA. And he came out with what we call Chargaff's rules, or the rules that every single piece of DNA seems to follow. He found that the amount of purine is always equal to the amount of pyrimidines. So A's and G's, if their A's and G's are 50%, then cytosines and thymines are going to be 50% of your sample. And he found other relationships. The amount of A is always equal to the amount of T. So the amount of adenine is always equal to the amount of thymine in a DNA sample. So if I have 10% adenine, I have 10% thymine of my DNA sample. This would be a strange low number, but he found the same relationship between cytosine and guanine. If there was 40% cytosine, then there is 40% guanine. The amount 50 and 50 here, 50% adenine, 50% purines, 50% pyrimidines, but the A's are equal to the T's, adenine is equal to thymine, and cytosine is equal to guanine. Now, Chargaff then published his paper imagining that DNA looks something like this: AAAA, TTTT, GGCC, GGCC, GGCC. He thought still that the ability of DNA to send information was in its shape, not in the individual code of the nucleotides. Remember, people were coming from the idea that DNA was made out of 25% of each one of these nucleotides. He showed, one, that was not true. There were varying numbers of nucleotides in each DNA sample. It wasn't always 1/4, but also that some of them were related to each other. Meanwhile, all over the world now, people are trying to figure out the structure of DNA because we've accepted Hershey and Chase's experiment in the late 40s that DNA is the transformative molecule. Now we need to know what it is. And this is post-World War II, recently post-World War II. A lot of labs were switching over from doing wartime studies to more understanding how the world works studies. Rosalind Franklin was returning back from a fairly successful career on chemical weapons, treating people for chemical weapons, not developing chemical weapons. And she had skills in a process called X-ray crystallography. There's a Broadway play about this. There's a lot of background. If this is interesting to you, I can send you down a rabbit hole. Franklin really definitely took this picture of a form of DNA called bDNA that has this classic DNA structure that you're all familiar with, and we draw when you talk about DNA, this double helix. When she took this picture, that's what we're looking at here. Those are nucleotides, and they're crossing over in the middle. She's taking a picture of the helix there. Now, this was not the first picture she took. This is very dangerous. Actually, we know now the process is mutagenic for the researcher. She developed breast cancer and died young probably from doing this kind of research, but who knows, wartime stress and everything else. She did find out that the DNA itself is helix, and that helix structure is double helix. The width is consistent. I didn't give you the numbers here, but this width is always the same between twists. The width here, I'm not a good artist, but the width between the molecules making up the two sides are consistently. And the nucleotides themselves, the A's and T's and G's and C's, are stacked in the middle like rungs of a ladder. Very much like two twisted staircases, like a really fancy place twisting around each other. And no, I tried to find another one. This is a double twisting staircase. That model that she did, she was actually getting really sick. Her graduate researcher took it and shared it with Watson and Crick, who were working on building a model out of the molecules that you use for organic chem. And they're not really Tinker Toys. They're structurally recognizable atoms and molecules. When they built this model, they won the Nobel Prize for the model. Their paper is actually published in the same science journal, the same month, the same actual edition as Franklin's paper on the X-ray crystallography. Franklin died before the Nobel Prize was awarded, so she didn't win the Nobel Prize. Her grad student, however, did. So Watson, Crick, and Wilkins, who was Rosalind Franklin's grad student, won the Nobel Prize in the 1950s, 1953, for understanding the structure. She had actually died since then. The structure is important because structure shows us function. And the structure here that they built, because it was stable, allowed us to see how the genetic information was actually encoded. It's in the nucleotides and the nitrogenous bases, and how it replicates. This model, what we call the Watson and Crick model of DNA, is a double helix. It has a diameter of 20 angstroms, and every turn is 34 angstroms. When we look at a full, so this is one side and another side. I think for me, I had to look at this a little bit and actually start, there's some models I have for you that are physical models we can start looking at. The pink in this is a sugar phosphate backbone. This is the sugar is the deoxyribose. The phosphate is the phosphate group. Those are consistent throughout the entire molecule. Every nucleotide in the molecule has the sugar phosphate. Every nucleoside has that sugar phosphate backbone. Each sugar phosphate backbone nucleoside is joined to the next one by a phosphodiester bond, or a linkage. And this is a covalent bond that holds those particular monomers together. We're going to look at this a little bit more. You see how I say 3 to 5 here? The 3 to 5 tells me the direction of the sugar. It tells me the direction the molecule is facing. And so I will explain in more depth, but this is because we can't talk about up and down and left and right like I can in an art picture. In this picture, I can say this is up and this is down, right? But this molecule is floating around in solution. So the only way that I can describe orientation of the molecule to you is by describing the molecule itself and the shape of it. 3 and 5 are a shortcut for me. See? Here it says 5 and here it says 3. This 5 side is saying that this molecule is facing, this side of this molecule is the 5 side of this molecule. This side is the 3 side. We'll zoom in and look at it, but I want to show you here because it shows you on all of these diagrams that these two pieces of DNA that are twisting around in a double helix, held together by their rungs, which are made up of the nitrogenous bases, are running in opposite directions. This one, all the way here, is running from 3 to 5. And this one, if I trace it in the same way, you have to make this, you have to play the song too. Is running from 5 to 3. It gives me directionality of my molecule. So I'm going to reveal this. My little block is hiding. So we have 2 strands that make up a single DNA molecule. I flattened it out here for you, so it's not twisting. It's hard to see when it's twisting. They run in different directions. So if I drew a line down the middle here, this side is running 3 to 5. This side is running 5 to 3. They're held in the middle. These are the rungs of the ladder, which you would put your feet on. These are the rungs of the ladder. They're held together by hydrogen bonds holding the nucleotide, the nitrogenous bases together. There are 2 hydrogen bonds that hold the adenine and thymine together. There are 3 that hold cytosine and guanine. Hydrogen bonds are sticky. They're between molecule attractions. They're easier to break than the covalent bonds that are holding the sides of the molecules together. The 2 strands are running anti-parallel. They're parallel, but running in opposite directions. One is running 3 to 5, and one is running 5 to 3. So if you look, this side and this side are upside down versions of each other. That means that the ends, I'm going to go here, if the ends of these 2 molecules are running in opposite directions, they differ. The 3' end is the flat end. Here, this is 3, 3, 3, 3. And it's really referring to the fact that this is the 3rd carbon. 1, 2, 3 carbon there. The 3' end, boop, I'm going to highlight them all red right now. The 3' end has a free hydroxyl on it, a free OH, we will be able to use this for dehydration synthesis to build a new nucleotide right there. Or right there. I cannot build here or here. There is no free hydroxyl for me to do dehydration synthesis. The molecule that is doing the building cannot build on this part of the process. There is a phosphate group which is acidic and charged there, and that kind of keeps it from being a good place to build. This is the 5' side. That's because this is the 5th carbon in the molecule that is the sugar. We're saying what carbon is it closer to. The 5' end is closer to the 5 carbon of deoxyribose. The 3' end is closer to the 3 side of the deoxyribose. You can only build on the free 3 if you're elongating the molecules. Free 3. And I remembered that because it's really hard to say free 3. Just keep calling it the free. I can't even say it now. Free 3. So this is the gist of what is going to happen. We are going to take our DNA molecule, not in this lecture, not in another lecture. We're going to take our DNA molecule. We're going to pop apart the hydrogen bonds, but not the phosphodiester bonds. We are going to use a whole bunch of enzymes to build new strands matching base pairs to what they're matching hydrogen bonds. A and T each have 2 hydrogen bonds that they form a connection with. C and G have 3 hydrogen bonds, and we'll end up with 2 new daughter strands. Maybe let's put them together on a centromere here. 2 new daughter strands. This is S phase of interphase. We're just going to take the molecule apart and read the template and build new daughter sides. But again, that's another lecture. So there are alternate versions of this. DNA modeling and our structure is based on the Watson-Crick model, which was based on bDNA. That's what Franklin took a picture of, and that's probably what you imagine. Luckily, and I don't even think luckily, she probably got lots of pictures of different versions of DNA, but bDNA is the one that's actually easiest for us to model. aDNA is a little bit more compact than bDNA. It's if I have a lot of dehydration or high salt concentrations, it'll compact down. It's dehydrated. C, D, E, and pDNA are right-handed forms instead of left-handed forms. They are less compact. They're more unraveled. I don't have too much for a very long time. I don't know if we're ever going to get to it. I think I would be very happy if you know that DNA does not always look like the picture that I showed you. But it will for all of our explanations in the next couple lectures. RNA has a different structure than DNA. It's a different molecule that does different things. We're going to replace that deoxyribose with ribose. Ribose has not been deoxied, so it has its OH still there. We're going to replace thymine with uracil. Uracil is actually less stable than thymine. It, in solution, easily converts into cytosine, which kind of messes up the message a little bit if you were using it to code for you importantly. It's good for a temporary set of code, but not for keeping code for a lifetime. Most RNAs are single-stranded, but some of our animal viruses have double-stranded helices of RNA. There's a lot of variability in the structure of it. I have 3 types of RNA for you. I literally took these pictures from Biology for Kids. It's like a 6th grade textbook. For today, it's enough to get you the structure of the molecule. We will obviously look at it in more depth, but I like this as building the frame for what is going to be a very complicated, informative molecule. Messenger RNA is 1 strand. This is the phosphate sugar backbone and its variable nitrogenous bases here, including uracil. This is mRNA that is transferred from the DNA with its little bit of code to be taken to the ribosome for protein production. The ribosome itself is made out of partially rRNA or ribosomal RNA. It's made out of proteins and ribosomes working together. Sort of an interesting side note here, the ribosomal RNA is acting as a ribozyme. It is an enzyme that can do things, even though it's made out of RNA. Most of our enzymes are protein-based, but this one that we use still in our cells today is old-fashioned. It's a ribozyme. And tRNA, the most forgotten of all of the RNAs in Bio 1 students, tRNA transfers the amino acid by reading the message in the messenger RNA and by being catalyzed by the rRNA to build a polypeptide under the DNA's instructions. Transfer RNA is often forgotten because we practice with those little charts and we build our own amino acid sequences. So you read the message in the chart when you're practicing and you build the final polypeptide, but you are the tRNA in that story. So it's easy to forget that these molecules can't do that process of reading the chart and choosing the amino acid. That's tRNA's job. You're doing that in the process of practicing. So a couple little things. I want to talk about sort of analytical techniques. I chose some of them from the text and not the others because I think these are ones we'll talk about more. Depending on what we end up talking about towards the end in people's presentations, I'll bring those back and talk about them later if they're necessary. Molecular hybridization allows us to take two different DNA molecules, denature them, so that separates their hydrogen bonds, the rungs separate away from them, and then renature them as we control for the temperature in ways that we want them to bind back together. We're going to do a little bit, something similar to this in PCR. When we start to bring in, and after spring break, we're going to do a process where we use a thermocycler to control the temperature. It's a machine. Control the temperature up and down and bind DNA back together in ways that it hadn't been bound before. We also have fish. No, guy. No, not that kind of fish. No, get out. No, go. No, this is fluorescent in situ hybridization. It's much less exciting. We use fluorescent probes to look for hybridization, to see if my molecules are binding together the way I think they're binding together. I'm using, I didn't define it, single-stranded. And then I hybridize, monitoring it I with a glowing tag attached. In situ means in place. So I'm actually looking for this within a cell as a cell is living its life and doing things. The probes in this case, when we talk about fluorescent probes, they're nucleic acids, which are strands of nucleic acids that have a glow-in-the-dark fluorescent tag tied to them. They only hybridize with specific areas, so I can tag specific areas by having them grab the thing that they're most likely to bind with, and then they glow so I can see them with either my eyes or in the lab I had worked at in college, we had a machine that would highlight them and amplify them because it wasn't enough for us to see with our eyes. And finally, something we will do, depending on how the P-Glow experiment goes, we'll either set this up before or after spring break. We're going to do gel electrophoresis. And it does use an agarose gel. We're going to mix them and prepare them ourselves, sort of the same way we prepared the agar plates. We put the nucleic acids on an agarose gel. We submerge that in salt water, which is good at conducting electricity. Then we put our samples, I can't remember what order I said this in, we put our samples of DNA that we're going to prepare. We turn the machine on. It runs electricity through the process, and when it does that, the agarose itself acts as a molecular sieve because electricity causes the DNA or RNA molecule to wiggle. Because that molecule is charged and electricity is negative, the molecule itself is negative. The negatives move away from each other, so we'll move the DNA through, or the RNA through the sieve, as it moves, it gets caught up early if it's large. It can move further if it's smaller. It separates them by size. Then we don't use ethidium bromide in our labs, but you either use ethidium bromide or some other glow-in-the-dark substance, and we'll turn on a UV light and look at our DNA as bands and try to see what we got. It does not let us see the A's and C's and G's and T's, but it does let me see the difference between one gene and another, and it lets me see the difference between my not normal gene and your normal gene. If my gene is not normal, it often means there's been an insertion or a deletion or some sort of change to the molecular code that affects length of that segment. Genes that are supposed to be the same that are different lengths are different alleles, and I can actually look at the different alleles by separating them by size. We'll do it. This is always weird when you first think about it, and then after you do it, you're like, oh, it's so simple and elegant and beautiful, and can actually be done in your kitchen. Mostly. I have some practice with me. These are very much like the practice on the homework. What's not consistent with Erwin Chargaff's findings? I'll give you a second, or pause me. He knew that purines and pyrimidines were found in the same amounts. He knew that A and T and C and G were found in the same amounts. A and T are found in the amounts equal to each other, but it doesn't mean that this purine-pyrimidine combo is the same frequency as this purine-pyrimidine combo. You can have 20%, that was the same example I used when I talked, 40% A's and T's and 60% G's and C's, or 10% A's and T's and 80, wow. 80% C's and G's, but they're not going to be necessarily, and their ratios have nothing to do with each other, except they all have to total 100%. How about this one? Oh, it looks complicated. I like it. The virus 5-Chi, is this 5-Chi? It's 5X174. Sounds like a Borg. Infects Escherichia coli, so this is E. coli. Its base composition is as follows. Okay, don't make any assumptions here, except this is a bacteria. And it has DNA that follows the rules. What do you know about 5X174? I'll give you a second, and then I'll come back and do a starter and then another break. All right, so I'm going to go ahead. If you're not done, pause me. Chargaff said that in DNA, the amount of adenine is equal to what? The amount of thymine. So I would expect if DNA is coding, I would expect that my thymine and my adenine should be the same amount. That is not true here. In fact, these numbers don't relate to each other much at all. So this, to me, tells me that it's not in a double strand, because A does not appear to be binding with T because they're not found in the same ratios. If you still haven't solved it, is this code because the DNA is not in expected ratios? Does that mean automatically that RNA is the genome for this virus? And then here's the answer. I'm going to give you time to pause. It's single-stranded DNA genome. It's not forming binding pairs, but it is using DNA as its code. This is a code. It's just not a code in double stranding. Well, I guess I thought there were 3 questions. I guess there are 2. So to sum up, we're going to do DNA replication. That is what's coming next. We're going to do DNA replication in bacteria and eukaryotes, much more in depth than what you have done in previous classes. I don't know depends on who you had for micro. Oh, you all had Dr. Stewart. I'll double check with her, but I think probably about the same level you did in micro if you did the 204. I will highlight the stuff that I think is new if you have only had bio, and I'll double check with her for where we are going. Otherwise, prepare for that, and we'll start talking about exam 2, which is coming soon, and spring break, which is coming soon, and hopefully I will see you on this week's in-person lab, and the lab will work, and it will all be wonderful. I'll see you in lab or in the next video. All right, bye-bye.